One question: why is there such a stunning diversity of life? One answer: evolution. Charles Darwin's brilliant theory that explains how species adapt and change. It's been called the best idea anyone ever had. But there's one big problem. How does it actually work? Now, extraordinary science is answering that question. It is uncovering the hidden mechanisms inside creatures' bodies that can explain astonishing transformations, like how birds can evolve from dinosaurs, why a fish was once your ancestor, and above all, what makes us human. Right now on Nova, you'll find out what Darwin never knew.
Major funding for Nova is provided by the following: ExxonMobil, taking on the world's toughest energy challenges. And by David H. Koch. And
discovering new knowledge. HHMI.
And by the Corporation for Public Broadcasting, and by contributions to your PBS station from viewers like you. Thank you.
Additional funding by Promega Corporation. Discovering every day. Promega, the science of life.
And by the Wisconsin Alumni Research Foundation in support of the University of Wisconsin.
The tree of life on Earth is one of stunning diversity. "9,000 species of birds, 350,000 kinds of beetles, 28,000 types of fish, 2 million living species, and counting. And we are just one of them. But why is there such an amazing variety of animals? Why are there so many types of fish? So many different species of beetle? How did this extraordinary profusion of life on Earth come about? Today, we celebrate the man who would ultimately answer that question. Charles Darwin. He was born 200 years ago, and it is 150 years since he published the work that has become the bedrock of our understanding of life on Earth.
What Darwin wanted to understand was how you get this extraordinary diversity of life on Earth. He was spot on. He really nailed it.
Darwin's theory of evolution, his account of why species adapt and change, has been called the best idea anyone ever had. But even Darwin admitted that his work was incomplete. Vast questions were still unanswered. And the biggest question was how. How did evolution take place?
He didn't know any of the mechanics of that process. He didn't understand the physical forces that would actually change the way species appeared.
But today, we can answer the questions that Darwin could not. We can look under the hood of evolution and see exactly how this mysterious process gives rise to such astounding diversity.
What's incredible about this time and from a scientific perspective is we're going to be able to understand that diversity. And that just adds to the excitement. It doesn't demystify it, it makes it all the more magical.
And this is the magic and mystery of evolution. Over eons of time, a single species gives rise to many. An ancient fish evolves to become the ancestor of all four-limbed animals, even us. And one species, our own, develops a large and uniquely complex brain, enabling us to dominate the planet. This is the search for the answers to what Darwin never knew. Darwin began his love affair with nature when he was a child, just like many of his modern followers, including evolutionary biologist Sean Carroll.
Well, I developed my interest in animals the same way I think most biologists did, which is either going out in the backyard or going to zoos.
And anytime I got a chance, I'd flip over logs and look for salamanders and snakes and frogs and things like this. And I was just fascinated with their patterns and behavior.
So it was with the young Charles Darwin.
Young Charles liked to traipse around the outdoors. He loved to collect beetles and things. He was a completely ordinary kid, and he didn't like school. In fact, he was such a poor student that his father, a rather successful physician and a pretty imposing figure, was worried about Darwin's direction in life.
So his father packed him off to Edinburgh, the finest medical school in Europe, to become a doctor. But young Charles was just too squeamish.
And he's really horrified by medical school. He witnessed an operation on a child, and this is in the era before anesthetics. And he just fled the operating theater, vowing never to return.
Next, his father sent him to Cambridge to study for the clergy. He didn't succeed at that either, but he did find his direction in life, reviving his childhood interest in nature.
Darwin starts on his path to his divinity degree, and he starts to mature as a student. He becomes more serious about some subjects, particularly natural history, and he learns a lot more about botany and about geology and these things. He's becoming a pretty solid field scientist.
His reputation as a naturalist gained him a spectacular invitation.
Charles was offered a place on the British Navy ship, the HMS Beagle, whose mission was to survey the waters around South America. Now, the captain of the Beagle wanted a well-educated scientific person aboard and a dinner companion, somebody to share conversation with. And Darwin fit the bill perfectly.
And so, Charles Darwin set off on a fateful voyage that would revolutionize our understanding of life's great diversity. The voyage of the Beagle took nearly 5 years. It wove its way from the Cape Verde Islands and along the coast of Brazil. It was in Argentina that he made his first important discovery.
Early on in the voyage, Darwin found some amazing fossils. He dug up some skulls, some jaws, some backbones of what turned out to be giant mammals. Now, these were clearly extinct. And Darwin began to ponder what was the relationship of those fossils to the living animals of South America.
But one port of call on Darwin's voyage proved more important than all the others. The Galapagos. This cluster of 13 isolated islands lies 600 miles off the coast of Ecuador in the Pacific Ocean. These islands are home to unusual animals found nowhere else on Earth. Penguins that live at the equator and swim in warm water instead of the frigid seas of the South Pole. Giant tortoises that weigh up to 600 pounds. Iguanas, huge lizards that swim and dive in the sea. Everywhere else, they dwell only on land. Traveling for the first time in the Galapagos, Sean Carroll is seeing the same creatures that so intrigued Darwin.
Of all animals, I think these marine iguanas are the greatest symbol of the Galapagos, what I most wanted to see here. And to see them in their native habitat blending against that black rock, just as Darwin described it, it's an absolute thrill.
It's a hideous-looking creature. Of a dirty black color, stupid and sluggish in its movements. They are as black as the porous rocks over which they crawl.
Darwin meticulously described the iguanas in his diary, but he was far from the scientific authority he would become.
The Darwin that arrived here was not the great theorist that we know today. He was a 26-year-old collector, collecting really almost at random any kind of plants, any kind of animals, any kind of rocks. He didn't even know the meaning of what he was collecting until much later.
He was also fascinated by the giant tortoises, which allowed him to ride on their backs as they slowly lumbered around.
I frequently got on their backs, and then, upon giving a few raps on the hinder part of the shell, they would rise up and walk away. But I found it very difficult to keep my balance.
Darwin measured the creature's extreme slowness, about 4 miles a day, he calculated. But the local people knew something else about the tortoises.
That they could tell which island any tortoise came from just by looking at its shell.
Their shells differed depending on which island they lived on. Some tortoises had shells shaped like a dome. Others had shells arcing over their heads like a saddle. Others differed subtly in color, or by how much the bottom of the shell flared out. Darwin had literally been sitting on a clue, a way to understand the great diversity of life, but he didn't yet realize it. Instead, Darwin turned his attention to birds. The islands were full of what seemed to be a familiar assortment of species. So he stuffed his collecting bag with what he thought were types of finches, grosbeaks, wrens, and blackbirds. And then, after 5 weeks in the Galapagos, Darwin and the Beagle went to other ports in the Pacific, and finally set sail for home. On board, he started to sort through the vast number of specimens he had collected on the 5-year voyage. But it was not until he returned to Britain that he was able to make sense of them. It began with a startling revelation. All the different birds he had collected actually were variations of a single type.
He learns that those birds he had collected on the Galapagos actually represent 13 different species of finch.
What misled Darwin was that they looked radically different. Some had wide, tough beaks, others had long, slender ones. And these differences depended on which islands they lived on.
Now, why would that be? Why would there be slightly different birds, slightly different species on different islands all in one part of the world.
Darwin now thought back to the Galapagos tortoises. They too differed from island to island. His brain began racing.
Thoughts are starting to crystallize, take shape in his mind, bit by bit, bit by bit. He starts this process he describes as mental rioting, just stream of consciousness, where he's jotting down note after note after note, thoughts as they occur to him. And finally, they converge on this one idea.
What Darwin now realized was that somehow, for some reason, species change. Originally, there must have been just one type of finch in the Galapagos, but over time, it had diversified into many kinds, with different beak shapes. The same for the tortoises. One type of tortoise must have turned into many kinds, with different shells, depending on which island they lived on. With this great insight, Darwin entered dangerous new territory. The standard view at the time was that God had created every species, and that what God had created was perfect and could not change.
But Darwin said, no, why would the Creator bother with making slightly different finches for each of these different islands that all looked alike?
The prevailing view just didn't make sense. But this was only the beginning of Darwin's revolution. He turned his attention to the fossils he had collected in South America. One was of a giant sloth. Another was of a huge armadillo-like creature. These animals were extinct. But little sloths still existed in South America, and so did smaller armadillos. What could this mean?
It dawned on him that they resembled each other, so what he had found in the ground were the buried ancestors of the living animals of South America.
So again, here was more evidence that species changed. Somehow, these ancient giants must have been transformed into the smaller creatures we see today. But what Darwin would later find out took this idea of how species change into a completely new league. In Victorian times, scientists routinely studied life forms at the embryonic stage. How these tiny forms develop from just a single cell into an entire creature has long been seen as one of the wonders of nature.
Watching a developing embryo is truly the most glorious miracle of nature. I mean, you know, no baloney.
What Darwin learned from studying the embryos amazed him. In snake embryos, you could see tiny bumps, the bony rudiments of legs. But these would never develop in the adult snake. Darwin wondered, were snakes somehow descended from animals with legs? He learned that whales, which have no teeth as adults, had them as embryos. Those teeth disappeared before they were born. To Darwin, it had to mean whales were descended from creatures with teeth. But human embryos provided the most startling evidence. Under the microscope, tiny slits around the neck were clearly visible. Exactly the same structures were found in fish. But in fish, they turned into gills. In humans, they became the bones of our inner ear. Surely, this showed that humans must be descended from fish. It's an astonishing thought.
I don't know about your ancestors, but mine included priests and, you know, the usual suspects. But the idea that all of us have fish in our family tree, I think it's amazing.
And so Darwin arrived at an astonishing conclusion, one that would become central to his understanding of the great diversity of life.
Darwin had this amazingly bold idea, the tree of life, that all species were connected. And what it meant was, if you go far enough back in our family tree of humans, you'll come to fish. If you go far enough back in the family tree of birds, you'll come to dinosaurs. So that creatures that don't look anything at all like each other are actually deeply connected. No one came close to having this idea before Darwin.
This seemed to be an explanation for the vast diversity of animals. Beginning with a common ancestor, over time, across generations, species could change dramatically. Some might add new body features. Others might drop them. Ultimately, one type of creature could be transformed into something utterly different. It's a process Darwin called descent with modification. But it all begged a question. Why? What was making creatures change? Darwin needed clues, and he found them in a very surprising place. Dogs. Big, small, fat, tall. The British have long been obsessed by them. It was a full-blown love affair in Victorian England. Even Her Majesty was dog crazy. That love affair still continues today, especially among scientists like Heidi Parker at the National Institutes of Health.
So one of the most interesting things about dogs is the kind of variation that you have. And we have dogs the size of groundhogs versus a dog like Zeppy, the Leonberger, who can get to be the size of a mule deer. If we had that kind of size variation in humans, we would have people running around the size of Barbie dolls.
In his day, Darwin knew this range of sizes hadn't come about by chance. Through a careful process of selection, dog breeders mixed different dogs with different physical traits to create new forms.
Darwin was intrigued by what he was seeing breeders doing with the domestic dog. They could select for individual traits like size or shape, and they could actually change their breed.
The Whippet, for example, had been developed to chase rabbits. It was created by mixing greyhounds for speed with terriers used to hunt small game. And then it hit Darwin. Was there a similar form of selection going on in nature, but without human interference? Could natural selection explain the great diversity of life?
It was brilliant. He took something very familiar and comfortable, for example, animal breeding, and explained that the same sort of thing was going on in nature, just at a little bit different pace and with no human guide.
But what could be carrying out selection in the wild? It was then that Darwin took a completely fresh look at nature. The Victorian view of nature was sentimental. Lambs lay down with lions. But Darwin's travels on the Beagle led him to a different view. For Darwin, nature was savage. Every creature was locked in a desperate struggle for survival, ultimately ending in death.
The scale of death in nature is absolutely horrendous. And sometimes it's not just that there's a lot of death, but that it's very unpleasant death.
But in all this brutal chaos, Darwin saw a pattern.
Darwin showed that nature was a battlefield and that everything was in competition. And this brutal battle, this war of nature, as Darwin described it, was actually a creative process.
The pattern that Darwin saw was that the creatures that survived were those best adapted to the specific environments they lived in. For instance, some could handle extremes of climate. Others were brilliantly honed killing machines, perfect for catching the available prey. Still others were perfect to evade those who might be hunting them. But how did this harsh view of nature explain the finches on the Galapagos, where Darwin observed that the birds on different islands had different beak shapes? Somehow, those different beaks must be helping the finches survive.
The finches of the Galapagos Islands have beaks of many sizes and shapes, and there's a reason for that. They use their beaks as tools. Now, if you think of the type of tool you would want to crush a seed that's very tough, but it's the food that you really like, you'd want a beak like this, which is the type of beak the ground finch has.
On an island where the only food is seeds that are hard to crack, a short, powerful beak will mean a finch will survive. But on another island, the available food isn't seeds, but flowers.
If you wanted to get into narrow spaces to get pollen and nectar that are very hard to get at, you wouldn't need a big, strong beak, you'd need a probing beak.
So on a different island, where you have a different food source, you have a different beak shape. And this pattern was repeated across the Galapagos. It seems that the finch's beaks had altered to fit the diet of each particular island. And that was how one original type of finch had been transformed into many. But how had these changes come about? Here, Darwin had another clue. He could see it in his own family. As every parent knows, no two children are ever exactly the same. Charles looked different from his brother Erasmus, even though they shared the same parents. Charles's children looked a bit like him and his wife Emma. But they too looked different from each other. That was something he called variation.
He realized that not every individual was the same, stamped out like a toy from a press, but there was variation.
Darwin realized that variation must be the starting point for change in nature. In any generation, the animals in a litter are never quite the same. And in the wild, such a tiny variation might make all the difference between life and death. Two penguins, for instance, might differ a tiny bit in the thickness of their blubber, a big factor if you live in extreme cold. In a harsh climate, the environment will select who will live and who will die. And slowly, Darwin suggested over many, many generations, these tiny variations would allow the fit to get fitter and the unfit would vanish. These variations accumulate, and eventually new species branch off. This is evolution by natural selection. It is one of the keys to how new species are formed. And so, in 1859, after years of painstaking research, Darwin finally published his masterwork, On the Origin of Species. It is still impossible to overstate its importance.
It was really a quantum advance in understanding. It shook people up. It changed the way people thought.
Gone was the idea that all species were created perfect and immutable, taken as an article of faith. In its place, Darwin provided a proper scientific theory based on facts and observation.
It is much more than the presentation of simply the idea of natural selection. It's a vision of how evolution by natural selection works.
150 years later, his theory has stood the test of time.
What's amazing is that Darwin got so much right. His ideas largely stay intact today.
But Darwin himself acknowledged that there were holes in his theory. He didn't actually know how it worked. What was happening inside a creature's body that makes it change? But now, at last, modern science is providing the answers through a hidden mechanism that Darwin knew nothing about. Arizona's Pinacate Desert is a harsh and brutal place, especially if you're a rock pocket mouse.
They're the Snickers bar of the desert. They really are. They're eaten by everything. They're probably eaten by foxes and coyotes and rattlesnakes, owls.
Weighing just half an ounce, this mouse could never fight off these large predators. Its best hope for survival is camouflage. Not surprisingly, its fur matches the color of the Pinacate rocks. But in some sections of the desert, the environment is different. Ancient volcanoes erupted, and now the desert is a patchwork of dark lava and light rock. But of course, a light mouse on a dark rock is easy pickings. So something has happened that Darwin might have predicted. The mice now living on the dark rocks have evolved darker fur. Those that stayed on the light rocks remain light. Michael Nachman was fascinated. How had this happened? To find out, he first needed to catch some mice. So with Sean Carroll, he visits a line of traps he set the previous night.
All of the dark ones have a white underbelly, and presumably there's no selection for dark on the belly because predators are coming from above.
This much Darwin could have done. Find some mice and compare the color of their fur to their environment. But Nachman can now do something that Darwin never could. He can look inside the animal's DNA. The study of DNA is one of the great triumphs of modern science. It has taken our understanding of how creatures evolve and develop to a level that Darwin could never have dreamed of.
The DNA molecule is one of the real secrets of life. It's a perfect system for storing the vast amounts of information that's necessary for building all kinds of creatures.
DNA consists of one long molecule, spiraling around in a double helix. That helix is in turn made up of 4 smaller molecules, called by the letters G, A, T, and C. DNA can be found in the cells of every living thing on Earth.
The thing about DNA that I think is remarkable is that the molecule itself is so elegant. With a small number of letters, you can say almost infinite words.
And that is the key. DNA is a code, and its double strand contains all the information to make living things grow and develop. Lined along each DNA molecule are arranged special sequences of this code that form our genes. Many genes get translated into proteins. And these proteins make the stuff of our bodies. One protein makes hair. Another makes cartilage. Others make muscle.
What makes DNA so amazing is that it just contains 4 letters. But all sorts of combinations of those 4 letters contains all the information for making all the creatures that are on the planet.
It's a gene that determines whether our eyes are blue or not. Another gives us freckles. Another gives us dimples. But DNA has one other vital quality. It doesn't stay the same. When a baby is conceived, the fertilized egg receives half its DNA from the mother and half from the father, creating wholly new combinations. It's why we look a bit like our parents, but also different. Another way that DNA can change is mutation.
Mutation is a critical ingredient in the recipe for evolution. Without mutation, everything would stay constant, generation after generation. Mutation generates variation, differences between individuals.
Mutations can happen as our DNA copies itself when our cells divide and our bodies develop. An A, for instance, can be replaced by a G, or a C by a T. This can cause minute changes that no one is even aware of. But when mutations occur in the cells we pass down to our children, they can cause big changes. Like turning a light-colored mouse dark.
Mutation seems to mean that something bad has happened. Well, mutations are neither good or bad. Whether they are favored or whether they are rejected or whether they're just neutral depends upon the conditions an organism finds itself. So for the pocket mouse, a mutation that caused the mouse to turn black, that is good if you're living on black rock, and it's bad if you're living out in the sandy desert.
It was that mutation, the one that turned a light-colored mouse dark, that Michael Nachman was hunting for. Back in the lab, he began the painstaking business of comparing the genes of the 2 types of mice, trying to pinpoint any differences.
Science is fun when you really don't know what you're going to find.
One by one, the genes in the 2 mice proved identical. But then, in one gene, he found something. There were 4 places where the sequence of A's, T's, C's, and G's were different. When a mouse is born with these mutations, its fur grows dark. And that means it can survive on the dark rocks when others would not. Here was a clear example of evolution and natural selection at work.
I think Darwin would have been delighted to know that we can find the genes that are responsible for evolutionary change.
And this was just one of many links that have been found between genetic mutations and evolution. Scientists can now pinpoint a range of examples of evolution in action. The colobus monkey can see in color because of a mutation in one gene. It can now tell nutritious red leaves from tough old green ones. A genetic glitch gave this Antarctic fish a potent antifreeze in its blood, so it can survive in the icy waters when others cannot. So powerful was this link between genetic mutation and evolution that an idea took hold. To understand how evolution works, all you need to do is compare creatures' genes.
One might think that you can understand all of evolution simply by mapping the genes of every creature. Identify all the genes, identify all the differences, and you could explain the differences between, say, mouse and monkeys and humans.
So when the Human Genome Project began in 1990, the scientific world was on tenterhooks. All 3 billion letters of our DNA were going to be identified in order. In parallel, the DNA of some animals and plants was also being sequenced. Surely this would be a quantum leap in our understanding of how different life forms evolved. With this came another idea, that complex animals like us would have many more genes than simpler ones.
Here we are, the most complex and sophisticated animal on the planet, right? You might think that would require a whole lot more genetic information.
The betting was on. Just how big would our genome be compared to other life forms?
There were estimates that humans would have between, let's say, 80,000 and 120,000 genes.
So when the final answer came in 2003, it was a shocker. 23,000 genes. The same number as a chicken. Less than an ear of corn.
I mean, people were freaked out by the relatively small number of genes. It's down to something like 22 or 23,000 protein-coding genes in the human genome. The simple nematode worm has about that same number. And there are plants that have considerably more genes than the glorious human genome.
The whole Human Genome Project has been a humbling experience, as we've discovered that actually it doesn't take as many genes to make a human as we'd all hoped.
And it wasn't just that we had so few genes, but many of our key genes were identical to those of other animals. Huge though the breakthrough had been, the genetic revolution had opened up a whole new set of puzzles. As a solution to the mystery of how evolution works, genes and their mutations were only part of the story. There had to be something else, more subtle and more mysterious going on.
We have to explain then, how do you get all these differences if you have really similar sets of genes?
The quest to uncover what Darwin never knew would have to start again. The first tantalizing clues would come from those life forms that Darwin himself had studied, embryos. Look at these embryos. It is almost impossible to tell just days after conception which is the chicken, the turtle, the bat, the human. They look almost the same. Only as they grow does it become clear which is which. Darwin wondered, as scientists do today, how could they start out so similar and end up so different?
There is something profound about what the embryo is telling us, and we have rediscovered what Darwin was talking about all along, that the embryo is where the action is in terms of animal diversity. It is the platform for diversity.
What fascinates modern biologists is that all these different animals don't just look the same. They are using virtually the same set of key genes to build their bodies. The body plan genes determine where the head goes, where the limbs go, and what form they take, whether they are arms, legs, or wings. Another set of genes determines an animal's body patterning, the blotches, the stripes and spots. It is the same genes at work in every creature, from the leopard to the peacock to the fruit fly. And yet they produce radically different results. This has led scientists to a crucial insight about how animal bodies have evolved. It's not the number of genes that counts.
It's not the genes you have, but how you use them that generates the great diversity of the animal kingdom.
Finding out just how these same genes are used to create such amazing diversity has been the work of Sean Carroll and an unlikely hero of modern science, the fruit fly.
As much as I'd like to study the mammals of the African savanna, they make poor choices for laboratory animals. They're large, expensive, and they reproduce very slowly. To get data, we have to find the simplest examples of the phenomenon we want to understand.
But the humble fruit fly does weird and wonderful things. This fruit fly is dancing for sex. A rapt female takes in the show. She's particularly besotted by the dark spots on the male's wings. Watching it all is an equally besotted Sean Carroll.
You might think them just be annoying, but they're really charming. And the males of this species does a rather elaborate courtship dance where he displays these spotted wings in front of the female. To us, is as magnificent as what a peacock does.
But in some species of fruit fly, the males don't have wing spots.
So there's another fruit fly species that's different from the spotted species in two important ways. It doesn't have spots on its wings, and it does a lot less dancing.
Here, then, is a classic evolutionary puzzle. Why does one type of fly have spots and the other doesn't? Sean Carroll wanted to know. What is going on in their genes that makes them different?
So we wanted to take apart the genetic machinery for making wing spots to understand how those wing spots evolved.
Carroll began the process of sifting through the two types of flies' DNA. He had one clue to set him on his way. He already knew the gene that codes for the black wing spots. He calls it the paintbrush gene. But surprisingly, when he compared the genes of the two flies, they both had that gene, and yet only one had spots.
When we look at that gene in the two species, really, they both have this paintbrush gene. So the big difference is not having the gene, it's how they use it. One species uses it in the wing to make spots. The other one doesn't.
So why did the paintbrush gene create spots in one type of fly, but not in the other? In search of answers, Carroll turned to one of the least understood regions of DNA, the vast stretches that were once known as junk. It has been called the dark matter of the genome. Mysterious, uncharted, strange. The vast bulk of the double helix, some 98% of it, doesn't code for proteins, which make the stuff of our bodies. The genes which do comprise just 2%. Even now, no one is sure what much of this huge non-coding area actually does, but it has long beckoned evolutionary detectives like Sean Carroll.
So, this is the band.
So that's the fragment to test.
Yeah.
Carroll had already learned that the paintbrush gene itself was identical in the two types of fly, so he extended his search through their DNA. And in one place, just outside the paintbrush gene, he found an important clue. A stretch of DNA that was different in the fly with wing spots. What could this mean? So Carroll conducted an experiment.
So, you injecting glutathione today?
He decided to put that mysterious stretch of DNA that he found in the spotted fly in the unspotted fly. To help him see if it had any effect, he attached it to a gene from a jellyfish, a gene that codes for a protein that makes the jellyfish glow.
We cut the DNA up into little pieces, and we hook it up to a protein that glows in the dark. And then we inject that into the unspotted fly.
And then something remarkable happened.
When we looked at those unspotted flies, we see now their wings are glowing in the dark with spots.
Somehow, that mysterious stretch of DNA had turned on the paintbrush gene in the unspotted fly's wings. Once spotless, now it had luminous spots.
Bingo. We'd found the piece of DNA that mattered.
Carroll had found something that is revolutionizing our understanding of how different animal bodies have evolved. A piece of DNA called a switch. Switches are not genes. They don't make stuff like hair, cartilage, or muscle. But they turn on and off the genes that do.
Switches are very powerful parts of DNA because they allow animals to use genes in one place and not another, at one time and not another, and so choreograph the spots and stripes and splotches of animal bodies.
In the case of the fruit fly, it's a mutation, a change in just a few letters of the DNA that has caused the paintbrush gene to be switched on. And so, a whole new species with wing spots has been created. But switches are now explaining far more than that. They are helping to solve many perplexing evolutionary questions, like how one creature can become another creature by losing its legs. It all goes back to what Darwin had seen in the snake embryo. The rudiments of leg bumps. This convinced him that a snake must have evolved from some four-legged animal. Over the years, that same mysterious process, the losing of legs, has been seen in other creatures, like the whale. Its front flippers have all the bones of a land creature's arm, even the fingers. And further back in its body, it has the vestiges of a pelvis. Clearly, it is descended from an animal that walked on the land.
Lots of animals have evolved to slither through the ground like snakes. Other animals slither or swim through the water like whales. So if you need a streamlined body, it's good to get rid of these things that stick out from the body, like limbs.
Like the whale, the manatee is another huge mammal that lives in the sea. And it too has lost its hind legs. How? Darwin could never have answered that question. But now, thanks to our understanding of how DNA is switched on and off, and a very small fish, we are getting a little closer. In this lake in British Columbia, there's a creature that really shouldn't be here. A stickleback. Most sticklebacks live in the ocean. But some 10,000 years ago, a few were left stranded in this lake, cut off from the Pacific. And over the years, they have evolved. The ocean stickleback has a pair of fins on its belly that are like spikes. They are for defense